Don't be discouraged if this video doesn't seem to make a lot of sense right away. Remember that the normal expectation is that you would watch any video 3 times. Watch it once through from the beginning to the end, just to try to get a sense of what's going on. Watch it again and take that opportunity to copy down what you see on the screen and make yourself a really good set of notes. And then watch it a 3rd time, both to make sure that your notes are complete, but also to help you to remember to jot down any questions that you have about the video for the next time we see each other in class. Also, it's probably going to take 2 or 3 class periods of activities before you start to feel like you really have a handle on what it is that's going on with the structure of DNA. So don't feel discouraged if it feels pretty fuzzy even after you've watched this video 3 times this evening. First, DNA is a short form that stands for deoxyribonucleic acid. We call it an acid because if we dissolve it in water, the resulting pH is less than 7. We call it nucleic because we only find it in the nucleus of a cell. And we call it deoxyribo because one of its major components is a type of sugar that's called deoxyribose sugar. So between the fact that it's made of deoxyribose sugar, it's only found in the nucleus of a cell, and if we dissolve it in water, the resulting solution has a pH of less than 7. Those 3 things together give us the name deoxyribonucleic acid, or DNA for short. DNA looks like a ladder that has been twisted into a corkscrew. Have a look at these artists' renderings, these artists' impressions of what DNA would look like. Notice that there are 2 uprights, one on each side of the ladder, but that ladder is twisted into a corkscrew shape. Also, notice that the ladder has rungs. So we've got uprights, which are often called the DNA backbone, and the rungs that join the uprights together. The uprights are made of very long chains of the deoxyribose sugar I mentioned a minute ago. And that is separated by phosphate groups. So it's a long chain of these little sugar molecules where each sugar molecule is linked to its neighbor by a phosphate group. That phosphate group is the same kind of phosphate group that you saw in your chemistry unit. The rungs of the ladder are attached to the sugar molecules rather than the phosphate group. Despite the thousands and thousands of rungs on each DNA ladder, the rungs themselves are made out of only 4 different molecules. They are adenine, which is represented with a capital A, thymine, which is represented with a capital T, cytosine, which gets a C, and guanine, which gets a G. All right, here's another way to represent a DNA molecule, and you're going to see quite a lot of these in the next few days in our lessons. Let's take a look at the components of the diagram. First of all, we have our phosphate groups. Our phosphate groups are linking together our sugar molecules. And you'll see that that's happening on both sides. So the phosphates and the sugars make the uprights of the ladder, or the DNA backbone. And then there are rungs, and the rungs are what joins the 2 backbones together. Copy this labeled diagram into your notes, please. Also, add a note that adenine, which is the A, always pairs with thymine. And we see that all the way down this sample chain. A only pairs with T, and T only pairs with A. In fact, you can see in the diagram that we've tried really hard to emphasize that fact by making sure that the A and the T fit together. They've got complementary shapes. Likewise, cytosine, which is the letter C, given the letter C, always pairs with guanine, which is the letter G. And we see that all the way down the chain as well. So the C and the G also have a complementary shape. So the G is sort of a forward bulging arc, where the C is an inward bulging arc. But those are rounded shapes, so those aren't going to pair very well with either the A or the T. And we've done this very deliberately to try to make it as clear as possible that only A and T will bond together, and only G and C will bond together. You will never get a G bonded to an A, for example. Finally, the two halves of the ladder are joined in the center of the rungs, and this is the weakest point in the DNA ladder. It turns out to be a really good thing that this is a weak point in the ladder, but we won't discuss that for another few days yet. We'll come back to this point when we talk about how the DNA molecule can copy itself. But that's everything that you need to know for tonight.